Adaptations of muscle to exercise. An athlete trains the various physiological systems to encourage adaptation and to improve performance. This training must be specific to the desired outcome, since the body can be subjected to large variations in exercise intensity and duration. At one extreme, resistance training can involve very heavy loads with minimum repetitions. At the other, long-distance cycling or running requires very submaximal muscular effort, but it is extended over a period of time. Because of this large diversity of possible exercise stimuli, adaptations in muscle tissue are similarly variable. A given activity dictates what type of muscle fibers, the subcomponents of muscle, each with differing structural and functional characteristics, will be recruited. This section discusses the muscular adaptations to strength, such as Olympic weightlifting, hypertrophy, bodybuilding, and endurance aerobic training. For in more depth, refer to Chapter 1 and 8. Muscular growth. The muscular enlargement that results from resistance training is called hypertrophy and is primarily a result of an increase in the cross-sectional area of existing fibers. The process of hypertrophy involves both an increase in the synthesis of the contractile proteins actin and myosin, the primary components of the two respective types of myofilaments within the myofibril, and an increase in the number of myofibrils within the muscle. The new myofilaments are added to the external layers of the myofibril, resulting in an increase in its diameter. These adaptations create the components or the cumulative effect of enlarging the fiber and the associated muscle or muscle groups. It is also theorized that an increase in the number of muscle fibers increases the overall size of the muscle. In this theory, initial muscle undergoes hyperplasia, longitudinal fiber splitting, as a response to high-intensity weightlifting. Hyperplasia has been found to occur in animals, but studies both reject and support hyperplasia in humans. Training programs. In general, the type or mode of exercise dictates the changes in the muscle or other connective tissue structures. As previously noted, the relationship is known as specificity. The three common and most general modes of exercise pertaining to the muscular adaptations are strength, hypertrophy, and aerobic endurance training. Training for strength. A strength training program is characterized by high resistance, near maximal muscle contractions for a small number of repetitions with a full recovery period between each set. Therefore, the relative intensity of an exercise is high and the total number of repetitions for each exercise is low. The type of training elicits an increase in the cross-sectional area of the exercise muscles, with type 2 fibers areas increasing more rapidly and at a faster rate than type 1 fibers. The degree of the rate of hypertrophy of type 2 fibers demonstrate their greater recruitment during strength training than during other modes of exercise. This is a desired response, since type 2 motor units produce a greater force output and contract with greater velocity than type 1 motor units. Additionally, an initial dominance of type 2 fibers is an advantage to increase muscular hypertrophy. Additionally, an initial dominance of type 2 fibers is an advantage to increasing muscular strength because their growth results in an increase in lean body mass. One of the most influential factors in determining maximal strength. The biochemical adaptations to strength training lie in the significant increase in muscular glycogen, creatine phosphate, and adenosine triphosphate, ATP, substrate stores. There is also an increase in the quantity and activity of glycolytic enzymes, myokinase and creatine kinase. The higher level of substrates requires additional enzymatic activity to speed reactions so that energy stores can be used efficiently. Training for muscle size. A hypertrophy or bodybuilding training program involves using moderate loads that allow for an athlete to perform more repetitions than is typical of a strength training program, but heavy enough to load to elicit concentric or eccentric contraction failure, inability of the muscle to shorten or lengthen under control within 6 to 12 repetitions. The rest period is of short to moderate duration because it is important to begin the next set of the exercise before full recovery has been achieved. Also, it is not unusual for the athlete to perform 12 to 20 or more successive sets that focus on one muscle group during a single training session. The higher overall training volume, coupled with a moderate relative intensity, pressed as a percentage of one repetitional max, or 1 RM, appears to be optimal for increasing muscle girth. Research indicates that bodybuilders exhibit a large absolute amount of collagen and other non-contractile connective tissue that also contributes to the increases in overall muscle size. Other cross-sectional studies revealed a lower percentage of type 2 fibers in bodybuilders than found in other anaerobic athletes, and a larger number of size of type 1 fibers. Note that some of these characteristics are similar to those found in aerobic endurance athletes. The biochemical adaptations to hypertrophy training programs are similar to those resulting from strength training programs, and both regimes result in type 2 fiber hypertrophy. There is discussion, however, that the increase in creatine phosphate levels do not benefit maximal strength training activities because the duration of actual exercise, i.e. the set, is too short. Increased creatine phosphate levels could possibly influence the total power output that can be extended over the 20 to 30-second duration typical of a higher repetition and lower resistance bodybuilding training programs. Training for muscular endurance using aerobic exercise. The muscular components of an aerobic endurance training program involve submaximal muscular contractions extended over a large number of repetitions with little recovery allowing between each set. Therefore, the relative intensity is very low and the overall volume is very high. This manner of training encourages relative increases in aerobic power potential that are similar in type 1 and type 2 fibers. Because, however, type 1 fibers have a higher pre-existing initial aerobic capacity than type 2 fibers, to which the increase in aerobic potential from training is added. Thus, type 1 fibers possess an oxidative capacity greater than that of type 2 fibers, both before and after training. Whereas strength and hypertrophy exercises resistance training produces somewhat similar muscular adaptations, aerobic training adaptations are different. Aerobic endurance training reduces the concentration of glycolytic enzymes and can reduce the overall muscle mass of the hypertrophied and non-hypertrophied type 2 fibers. Conversely, there is a selective hypertrophy of type 1 muscle fibers due to their increased recruitment during aerobic endurance activities. Although this resulting cross-sectional diameter is not as great as seen in type 2 fibers after adaptations to resistance exercise. Also, the change is similar than the hypertrophy of type 1 fibers from a bodybuilding style of resistance training program. There is little evidence to show that type 2 fibers change into type 1 fibers as a result of chronic aerobic training, but there may be a graded conversion of type 2B fibers to type 2A fibers, the two major type 2 fiber subgroups. This adaptation is significant in that type 2A fibers, or fast oxidative glycolytic (FOG) fibers, possess a greater oxidative capacity than type 2B fibers, also called fast glycolytic (FG) fibers, as well as functional characteristics more similar to type 1 fibers. The result of this conversion is a greater number of muscle fibers that can contribute to aerobic endurance performance. At the cellular level, muscular adaptations to aerobic endurance exercise include an increase in the size and number of mitochondria and a greater myoglobin content. Mitochondria are the organelles in cells that are responsible for the aerobically produced ATP via oxidation of glycogen. When the larger and more numerous mitochondria are combined with an increase in the quantity of oxygen that can be delivered to mitochondria by higher levels of myoglobin, a protein that transports oxygen within the cell, the aerobic capacity of the muscle tissue is enhanced. This adaptation is further augmented by the increase in the level and activity of the enzymes involved in the aerobic metabolism of glucose, and a parallel increase in the glycogen and triglyceride stores.